On the agenda today is to explain the purpose of today's demo. For the wet lab portion of this, we're going to prime, inoculate, and set up a biofilm and image acquisition. We'll do a brief explanation of the BioFlux and how the BioFlux can enhance your biofilm research, and then look at data analysis and review this. The objective is to grow a primary oral biofilm and to evaluate the effect of various toothpaste, including Crest 2 from Korea, a Japanese variety called Access, Tom's of Maine, and Vitamins All Natural Activated Charcoal. There's been a lot of speculation about whether or not natural ingredients work for toothpaste, as there's a lot of hysteria happening, well, it's been happening for some time, talking about all the toxic things that are in toothpaste that will accumulate in your liver and your kidneys over time and cause cancer and other things. Of course, most of us know this isn't true. So what's in toothpaste? Arguably, the three most effective things in toothpaste are the top three here. A detergent called SDS or SLS, or sodium adenyl sulfate, has about 50 different names to it, but they all mean the same thing. It's simply a detergent. Sodium fluoride has been shown to have anti-biofilm activities, though the original intention for sodium fluoride was to enhance tooth enamel. And then abrasives that go along with the brush to help break apart things that might be going in your teeth. The toothpaste was diluted with 3 mils of TSB per gram, and the charcoal was diluted at 6 milliliters per gram, simply because it was anhydrous and the toothpaste was already in a liquid solution. The slurry was centrifuged at 2500 G for 5 minutes to separate out the abrasives from the salt, herbs, sodium fluoride, and detergent. The experimental plan today is to inoculate primary saliva, which I've already done, and then we're going to prime that into, or seed that into a plate, into our 48-well 0 to 20 dyne plate. Going to inoculate this for an hour, which at least do in principle, and then look at adding the diluted toothpaste in media as a growth culture, and then imaging for a series of a time lapse. Limitations to today's demo: TSB and other medias are really not physiological. The classic media for strep mutans, which is a classic, one of the early biofilm bacteria, is to use BHI, brain heart infusion. But really, saliva is the gold standard. And one of the key advantages to the BioFlux is that it enables low volume, so saliva is feasible. The reason I didn't do that for this experiment is that dissolving toothpaste in TSB is hard enough. Dissolving toothpaste in saliva is just, I didn't want to spend my time doing that. So in this experiment, we're going to look at continual exposure, meaning that after the bacterial culture is seeded down, they'll be exposed immediately to the treatment conditions. Another option in a follow-on experiment would be a pulsed exposure after you've grown and developed the biofilm, to go back and then expose it to a treatment condition. The isolates came from my mouth. Mixed cultures do act differently. And one of the other things to consider is that the surface that we're going to be working with is glass. Some bacteria and some people would prefer that the surface be coated with proteins that may be found in saliva before starting. So I'm going to now switch to the video mode to show you how we'll start the wet portion of this laboratory. I'm going to start out today by inoculating 50 microliters, or priming the channel, I'm sorry, with 50 microliters of media. So we have our BioFlux software here. And hopefully you can also see the videos on the other side. In the inlet, I'm putting 50 microliters directly into the middle of the plate, making sure to avoid air bubbles and to avoid pipetting onto the side of the plate, which would mean that the liquid wouldn't actually flow into it, but would just perfuse air instead. I'll attach my interface. With the BioFlux 1000, we have a clip-on interface like this. With the 200, we have a screw-in interface. I'm going to run this in manual mode, and I'm going to perfuse at 1 dyne for about 5 minutes here. So while I'm perfusing, I'll show the type of toothpaste I'm going to use. This is Tom's of Maine, if you can see that. The interesting thing about this toothpaste is that this is all natural, but actually contains SDS, contains sodium fluoride, hydrated silica, and I think the only thing actually natural on it is peppermint oil. I've got 3 foreign toothpastes here. This one is called pine salt. This one's called bamboo salt, and those are both from Korea. This one's called Access, that's from Japan. And I've got something here from our logistics manager who wanted to try out this activated charcoal from Vitamin, and that contains a whole host of things, including bentonite clay, activated bamboo charcoal powder, organic orris root powder, stevia powder, hibiscus flower petal powder, and organic peppermint oil and organic cinnamon extract. So there are a couple ways to tell if the plate's primed, but the way I like to do it is to look on the back side here and see if a bead of media has formed. And we're going to give it about 2 more minutes before we go to the next step. We're going to pause it here. And before we go to add the culture, I think it's important to note that oftentimes people will grow up biofilm cultures, and unfortunately, they're probably forming biofilms before they go to inoculate that into the BioFlux plate. And that can lead to uneven loading. And if there's a portion of biofilm that would get pipetted in, it would cause some artifacts in the experiment. So 2 things that I typically do. One is to dislodge the biofilm with a pipette, and simply let's see this. pipette tip. And then the other is to use a strainer. Now, I've got a 40 micron strainer, which is used actually for like flow cytometry. And it may be better to even get something a little smaller, something like a 10 micron strainer. But very simply, we just add one to the other. And I can see on the strainer that there's some chunks of biofilm that have been lodged on this. Remove the interface. And I'm going to add 10 microliters, but now I can to the other side. The other thing that's important when adding this is to make sure that the suspension is still mixed up, because cells can clearly settle. I'm going to add the interface, or attach the interface. And I want to perfuse at 2 dynes for about 5 seconds, going from the outlet to the inlet. And then we'll check this on the microscope. See how the loading works. So this is in channel 2. We're running at 10x right now. And you can see that the material is loaded all the way to the end of the channel. This is where the material came from on this side. Now ideally, so I loaded about 5 seconds, and it clearly pushed it a little bit beyond where I wanted it. Ideally, you want the culture to stop close to here. If you go much farther than that, it's possible that the culture could build up and form a blockage in the serpentine region of the channel, and that would cause could cause a blockage potentially. The nice thing about oral biofilms is that they tend to grow in nice three-dimensional structures where the media will be able to perfuse around them. Some other cultures tend to grow in such a way that they would not, and they would just block themselves up. So hypothetically, this was sitting for an hour at 37. I like to incubate at 37 degrees simply because I think that the microbes adhere better to it. But I'm using this open style attachment here just so we can see this a little clearer. The heated insert is a little harder to see when you're adding liquids to it. So assuming that I've already done this for an hour, I'm going to add my media or dilutions. The most important thing to consider right now is that my media should be at 37 degrees if I'm going to run this at 37 degrees. If it's not, as the media heats up, it'll generate an air bubble, and that will cause a disruption to the flow. And this is the media. This is the Japanese toothpaste. The lab has certainly smelled very minty after we started doing this. This is the Korean toothpaste that has pine salt in it. This is the bamboo salt toothpaste from Korea. Any bubbles that may be on the top of the well won't be a problem. It's the bubbles that would lodge themselves on the bottom of the well that would cause an issue. This is Crest. And this is the Tom's and Me. One additional thing I'm going to do for this experiment is I'm going to add well, the eventual dilution will be 1 to 10,000, but this is a diluted stock of backlight, which is propylium iodide for dead cells and cyto9 for living cells. I diluted this 1 to 100, and then now I'm diluting, putting 5 microliters into 500, so that makes it 1 to 10,000. Another limitation to consider about this experimental model is that the toothpaste fractions that I've taken likely have a higher viscosity than the media. This is what the toothpaste looks like after being centrifuged down, that you have an aqueous layer and then a layer of solids. And so I simply took from the aqueous layer of this. So ideally, all of the detergent, the salts, and the sodium fluoride went through with that. Attach the interface. And put this onto the microscope. To set up a time-lapse acquisition, I want, under the multidimensional acquisition, time-lapse, multiple stage positions, and multiple wavelengths. Under time-lapse, I'm going to do this every 5 minutes for 18 hours. I think in my description I said 12 hours. It really depends on how long you want to run the experiment. And then I'm going to start with a new stage list. So I'm going to load I call it the 0 list here, which empties that out. And then as far as wavelengths, I want the FITC channel, the DS red, the CY5 just in case I catch anything that's off target, and then 2 bright fields. but I'm going to look at this in the bright field for determining the stage positions. So there was a question about whether I'm considering CREST as a positive control or if I have a no treatment control. So the media is the no treatment control. And I have some data I can show you on a positive control where I used 2% Triton. But yes, feasibly I would consider CREST also to be a positive control in that perspective. But as far as a negative no treatment, I'm using pure media. So I'm going to select 6 stage positions. I'm making sure that my X, Y, and Z are in focus here, or correct. So that's position 1, which is the media treatment. Position 2, which was the Japanese toothpaste. This is the serpentine, which I'll explain a little bit later. This is position 3. While I'm doing this, I can go ahead and start the flow, even though hypothetically I would have incubated this for slightly longer than I have. 5, and going from the inlet to the outlet. So here you can see material already starting to go from the left to the right. Position 4. Five. And 6. So I'll point out here that these areas here are accumulations of biofilm that fell through the 40 micrometer filter. So yeah, in the future it may be more prudent to do something like a 10 or a 5 micrometer filter with the idea of having a more even seating. I can go back and check these positions. And then I'll show you what acquisition would look like. This is cycling through the stage list. Given that there isn't much material here, the PITC and the DS red channels will actually where you see the larger amounts of material. They have already stained. And given that we don't want to stay here for 18 hours, I'm going to stop the acquisition once this is finished. I forgot to mention at the beginning of my talk that the most important thing right now is that there's a new episode of South Park premiering at 10 p.m. Eastern Time, and I guarantee you that this webinar will be finished by 10 p.m. Eastern Time. So I'm going to explain a little bit about the BioFlux, and then we're going to go into look at some data sets. Just a brief background about Fluxion. It was founded in 2005, headquartered in California, and we have distributors now over most of the world, including Australia and Africa. We had several NIH grants initially to fund the research, and we've got 3 product lines that have come out. What I'm talking about today is the BioFlux for live cell imaging. The IonFlux is a platform for automated electrophysiology or automated patch clamping. And the IsoFlux is a platform for enriching rare cells, the original use being for circulating tumor cells. So people often ask me, what's a biofilm? And the easiest way I can describe it is that if regular French fries were considered planktonic bacteria, the cheesy fry with bacon would be a biofilm. Perhaps thinking that the cheese is the extracellular matrix, and when you think about it realistically, when you get this Aussie fry from Outback, I'm sorry, only the American customers will really appreciate this, but you just can't get through it because it's impenetrable. And that's often the case with biofilms in the clinic as well. One of the original publications about biofilms came out from around the 1970s, where they described biofilms growing from wastewater. So there's often the question of why use flow for biofilms. And in this picture you can kind of get an idea that when you're using an agar slab, yes, you may be growing some biofilm and you'll be growing the bacteria, but you really lack the physiological conditions that you would see either in the environment, like in this stream, or in the mouth. And it was described back in 1932 that there was a case for both using flow and that there would be some sort of surface that a biofilm would grow on or bacteria would grow on. And this paper discussed that while there are bacteria in the water, most of them are growing on a submerged surface, so that they're not just free-floating like you would see in a shaker. So that's why the biofilm community really pushed towards laminar flow cells. The idea being that you had the component of a flow-based system and that you were able to have a surface that these would grow on. This type of system is effective, though there are significant challenges in sterilization, keeping air bubbles out, and for quantities of liquid that are used. So for the case of oral biofilms where you would be using saliva, to perfuse an oral biofilm using this type of apparatus, you would need to go across probably half of NIH to get them to spit into a tube for you. Whereas with the BioFlux assay using microfluidics, you're able to do this such that you may have 1 to 2 donors that could provide the entire study for biofilm formation. The advantages to the BioFlux is that it allows for a physiological representation of cell-based assays. It perfuses fresh media over just like a flow cell would, and essentially it's a coupling of flow cells, a multiplex of them. But what's more important is that it controls, or a significant important thing is that it controls shear flow. And while a peristaltic pump will be reasonably controllable by a knob, with an electropneumatic controller, you're able to precisely control the shear that's applied to this, but also but in addition to that, you can change that using the technology. It allows for temperature control, and it winds up being a cost-effective approach. So if you look at comparisons of approaches where flow cells are high on physiological relevance, they're typically low in throughput. And the idea that BioFlux has both a high throughput and a high physiological relevance. So there was a question that I missed a few minutes ago that asked, where the image is taken in confocal mode or with wide field. These images are taken with both in bright field and with epifluorescence. So these are not confocal images. The way that the BioFlux plate is designed, it allows for confocal images to be taken because the bottom of the plate is a number 1.5 cover slip. The BioFlux 200 system consists of an electropneumatic controller, a microfluidic plate, and software to drive the system. The 1000Z model that we have is what we're demonstrating today. Includes the controller, but it also includes an epifluorescent microscope with an automated stage and software to drive this. We use our own software called Montage, which is based off of Metamorph. The basic operating principle is that media goes into the inlet, is perfused through a microfluidic channel in the bottom of the plate, and comes out through an outlet. And based off of this principle of an inlet and an outlet, media and reagents can be pushed back and forth. Cells can be seated in a certain area. And at the bottom of the plate, again, is a cover slip. So an inexpensive, any kind of inverted microscope will work with this. An inexpensive one will be able to capture a basic image. A system like ours with an automated stage will be able to multiplex things. And a higher-end system, like confocal, will be able to take slices through it and have remarkable image clarification. The way the channels are laid out, we have two plate types. The 48 well has 24 assays that happen at one time, where you have on the left an inlet and an outlet. And so what you see here are actually two microfluidic channels. You've got two inlets on the left and two outlets on the right. And so this composes of two independent experiments. The application focus for biofilms include just growing simple biofilms, modeling assays, microbial adhesion, dose response, live dead assays, mixed cultures, which is what we're doing today. High content imaging is a possibility. Changing the microenvironment, so instead of perfusing room air through the microfluidic system, you can perfuse nitrogen or a mixed gas, and also evaluation of host pathogen interactions. These are some of the publications that have come out, and actually some of the early ones. We have quite a few more, including looking at Staph aureus, Staph epidermidis, mixed cultures, and a couple that are talking about, this is strep mutans from a oral biofilm group out of the 4th Military Medical Institute in Xi'an, China. Yeast, another Staph aureus, and I'm sorry, this is also another one out of the oral microbiology group at the 4th Military Medical Institute. And then a publication that came out from Bob Palmer's lab at NIH looking at microbial adhesion. And these are the groups that contributed to this work. There was a question about when the video will be posted for later viewing. Probably within about 4 hours after we finish. It'll be posted online. I'm sorry for the people on the East Coast. We picked this time because it was a compromise between being able to include the East Coast and be able to include Asia. And I did notice that there's some people from Spain that have actually gotten up at about 2 o'clock in the morning to see that. Oh, I'm sorry. I just got a message. It'll take a few days for them to get out all the ums from my talk. Thank you. The 24 well plate allows for 2 inlets to occur at one time. So you can have 2 different solutions that could perfuse either at the same time or alternating times. In addition to that, the reservoir volume for these is much higher. So if you want to grow a slower growing biofilm, like an environmental biofilm, you could use this to grow out your biofilm for about a week at a time. The 24-well plates allow you to perfuse in what they call laminar flow, whereas if you were perfusing a red liquid in inlet A and a green liquid in inlet B, you could see on the right that this would have a differentiation. So for early biofilm development and biofilms that form really smooth lawns, oral biofilms don't necessarily behave that way, you can do something with, in the same biofilm, looking at two different compounds where you would have a control on one side. This image is in the same biofilm using backlight. So the green is alive and the red is dead. And the top here is 1 ppm of hypochlorite or bleach. When you increase to 10 hypochlorite, 10 ppm of hypochlorite, you see that in the same biofilm, in the same channel, you've got a clear distinction between one treatment and the other. So this is really applicable for being able to look at the distinction between the two. Limitations with backlight, and we're using backlight today to evaluate this, is that PI stains DNA. So that includes extracellular DNA in addition to DNA and dead cells. Cyto9 can appear blurred. It's a great dye, but oftentimes it doesn't look as punctate as I would like. Typically GFP actually looks a little bit better. Probes, or Invitrogen, I guess now like Tech, has told me before that the live jet stain will only work as an endpoint, and that's usually if you call and tell them that things are not working. There are some microbes, including Candida albicans, that will not form a biofilm in the presence of dyes and UV light. So interestingly, you can apply UV light to C. albicans and get a biofilm. Say if you're doing red fluorescent protein or M-cherry and GFP, that would work fine. We have that video on our website. And you could add the dye and the biofilm would form, but the combination of the two is really interesting because you'll see a halo effect, or basically a hole where the light was hitting the biofilm, and that actually, or where the biofilm would have formed. And there's biofilm formed all around it, but not in that circle region where you've got both the combination of the dyes and the light. This is a study out of the University of Michigan where Alex Ricard was looking at concentrations of CPC, which is a chemical component in mouthwash, and titrating it out to see the killing effect with this. So in the top left corner, he used PBS, and you see it's mostly alive. Of course, a positive control of ethanol, which killed everything, and then decreasing concentrations of this compound CPC. He's later gone on to look at adding L-arginine, the amino acid, and magnifying the effect of CPC. He's got a patent pending on this, and I think the publication will come out soon. The classic biofilm model that is that you've got adhesion of the microbes, and then you form a structure, and then the microbes disperse. And the reason I want to point this out is that the oral biofilm tends to model really nicely with this, and so you'll see the biofilm form, and then it will roll off the plate. I've had some people before ask me, why did the biofilm roll off? Was there something wrong? And actually my response is typically, well, that's actually what a biofilm is supposed to do. So we're going to go and look at some of the data that we have here. So in the montage, this is for pre-existing customers or people who may want to reference this later, we would go to MM Standard, I go to Apps, and then Review Multidimensional Data. And stage position 1 was the negative control, or media with no treatment. And I want to look at FITC, DS Red, and one of the bright field images. And then I hit load images, which will load these into a stack. Okay. It's really not a good idea to close these windows while they're loading. That might cause problems with the software. So you can see from the beginning we have a small amount of seating. I'm going to make this window a little bit bigger for you to see. Small amount of seating that increases. This is in the bright field. Where I would call this a where that stage 4, where you've got the structure forming right before it disperses down and it goes and visits its neighbor. To evaluate the growth based off of the bright field, I'm going to threshold the image for dark objects. Let's say auto threshold. And take a look at this here, so I can regulate it. And then I'm going to scroll across and see that it's behaving the way I'd like it. The threshold is behaving the way I'd like it to. Unfortunately, we can't pick up every little detail in here. Otherwise, it gets into a question of signal versus background. But the majority of the large biofilm structures are being picked up. And take and put a rectangular region around this to avoid the edges, which have, of course, been picked up. And then to look at the kinetics of growth, I'll go to Standard, Measure, and Show Region Measurements. When this window opens up, sometimes it's montage behaving badly. So here you can see the logarithmic phase of where the biofilm was growing. And I'm going to change the threshold area to y-axis. I'd like to keep it 100% so that it doesn't auto-correct it each time and you can get a So what this is looking at is elapsed time, which is in milliseconds. You could always change that to seconds or minutes or hours if you want. And then thresholded area. So the percentage of this area that I've selected which has thresholded out. And you can see that the coverage goes to about 75%. And it takes about 9 hours for it to do this, roughly. So there's a couple things to consider when looking at what does this mean. One of those is the lag time between when you start and when active growth actually starts. You start the experiment and then active logarithmic growth happens. And then the other is the slope of how quickly it grew up. The length of, or the quantity of which it saturated, or the amount of biofilm that formed in the channel, and then potentially how much remained. But in the case of these ore biofilms, they roll off and then they come back into frame and they roll off, and so usually the usable amount of data typically ends right about here. I'm going to shrink this and take a look at the green channel. So this was the FITSI channel, where we used Cyto9 to image what should be the live bacteria. And take off the auto scan. Bring the histogram a little bit here. And you can see how the really starts about here. You saw you had that lag period. And this is running at about 4 frames per second. Again, this is my mount that was used to culture this. I'm quite proud of the organisms that grow, even though I do brush my teeth every day, and I visit the dentist twice a year. Okay. If we went to look at the, I'm going to turn off the pseudocolor because it's a little easier to see the thresholding. A threshold for light objects. And one of the nice things about using fluorescence is that it's a little more distinguishable than bright field. So I've taken, I have a little more signal that you can play with here. And I want to just make sure that I'm not, there's nothing crazy. Right about there, I don't like it. One of the questions I guess you could ask is if we could get a little more of this signal. Problem is that the microbes really aren't picking up the dye very well at this point, but very shortly after it crosses that threshold, and actually we have some points of saturation here, which that's probably why the auto threshold. Wait a minute, okay. So if we select a region, and then look at the multi-dimensional, or the region measurements. There's an artifact at one time point here, so we could always go back and remove that frame. But really what you're seeing here is the exponential growth of the viable portion of bacteria that comes across. And then if we look at the red channel, as I said earlier, the limitation to this is that extracellular DNA is also taken up and imaged. But thresholding this is really easy. Right about here. So some of this are the microbes that are dying as they go across, and the rest of it is the extracellular DNA. So there's some, a grain of salt to be had for, and we don't have any autofluorescence on the sides of the channel. Shrink that up a little bit. And this gives you an idea of the signal that comes through. That yes, it was there, but there wasn't much happening. The real dramatic change between live dead staining happens when you form the biofilm and then you decide to kill it later. Like we saw with, I can bring up that slide here, with Alex Ricard's work, where he took and established the biofilm growing, then later treated it with a compound. So he paused the experiment, treated it, and then imaged it, and essentially just took a terminal image. one of the things people ask me about is how can we make a video of this, and so we're going to look at putting together a video of the DS-Red contrasting, so the PI contrasting, the Cyto 9. So this is the propylium iodide. So we'll pseudocolor that to red. And the Cyto 9, we'll pseudocolor that, of course, to green. It comes out really nice. I'll try to take out some of the background. Then we have essentially two stacks, and we can go to image standard display, and then a couple options. You can go to overlay images or color combine. Oftentimes I'll try both of these to decide which one's going to be make a nicer image. So I'm going to go to the Fitzy stack here for one. I hit all planes, and then for the green I want to go to the DS-Red stack, hit all planes, and then hit apply. So this was run over 18 hours in this case, and you can see that so it references each frame, and each frame represents every 5 minutes that was taken. And this is what the video would look like if you were to put together a video for your lab meeting or for publication. Depending on how fast your microbes grow, you could get away with going significantly lower. And this can save as a TIFF stack, or if you'd like, you could export this out export this out into an API. So I know it's getting a little late on the East Coast. I'm going to go over some of the other sets of data that I have from adding in a different toothpaste. And I've already made the stacks, so you don't need to see me generate the stack each time. So I showed you what the bright field control looked like. This is an example of what, let this load up. Of what the bamboo toothpaste looked like from Korea. This is from LG. You can see that there's some material that's growing, but the majority of it doesn't adhere, and it's significantly attenuated. This is what we call the bamboo salt toothpaste. And as far as being in the fluorescent channel, you really don't see anything. Maybe a little bit of biofilm passing by, but not much. As a contrast, I'll open up a section of Crest, using Crest. So there was a question about whether we have a manual. We do have a manual online if you've registered your BioFlux to describe the image analysis. And this video will be available in a day or two. There's also a question about the number of well plates. So I used a 48 well plate where the inlet had 1 milliliter, consequently the outlet had 1 milliliter. That allowed for 24 assays. The 24 well plate allows for 8 assays. Right now we don't have a 12 or a 6 well plate, but we are developing a 6 well plate that will allow for an extended period of biofilm growth, particularly for things like tuberculosis. I usually just look at the bright field initially, which gives you an idea of any biomass that we're forming. And there is actually quite an interesting growth here that stays quite limited. And contrasting that to the control sample, the difference is quite dramatic. Then I'll show you here very briefly Tom's of Maine, which I thought was really interesting. Considering that it says it's all natural, but it looks like it looks like it's growing something. It's actually, it seems to be forming a lawn and keeping a lawn in this case. And then I want to open up this charcoal, which I mentioned before has organic hibiscus flower and other things growing in it. This one was really interesting because not only did it not inhibit biofilm formation, but actually the biofilm growing out of it looked a lot different than my mouth. If you can see these sort of structures that are forming here over time. I did try calling up this company to get a comment on whether or not this was a replacement for toothpaste, and they couldn't tell me. But as you can see here, there's something growing quite dramatically. And you can make a video to this as well. I'm going to open up one more video, and then I'm going to have to wrap this up because it's we're getting late. This is a different video, a different set I took a couple of days ago. And I'm using a 5x objective instead of a 10x objective, so we can see 2 channels at the same time. And at the top I've got the control toothpaste, or I'm sorry, control absence of toothpaste, and the bottom I have the pine toothpaste, the pine salt toothpaste from a Korean company. And I'll show you what the accumulation looks like. I don't have this in live dead. I just have it in bright field. But you can see the accumulation on the top versus the bottom. This makes a nice video to show people that they should be brushing their teeth. What I want to do is threshold for dark objects, and then modify the threshold image to reduce this background here. And then I'd like to isolate out two areas. So I'm going to take this one, and then I'm going to copy the region and paste it. I want the red one here and the yellow one here, but it doesn't really matter. Here and here. So I've gated out the negative control and my treatment group. And then I go to region measurements. So I hit all planes. And then by selecting on the yellow one, it shows the immediate growth of the biofilm. So in this case, the bugs were growing a lot faster than the other time. And as a contrast, here is the pine salt toothpaste, which is, you can see that it is barely going over top of the threshold as a function of time. So this is the threshold area, and this is the time lapse that goes along with it. One more to show you a positive control would be this a Japanese toothpaste contrasted with so the top here is the Japanese toothpaste and the bottom is 2.5% Triton, and you can see that there really is no growth at all in the bottom channel, or whatever it is, it's minimal. And actually, in this particular experiment, the Japanese toothpaste was higher than I would have expected. So at this point, I'd like to, oh, sorry, I have a few more questions here. So hopefully I've addressed, with this, looking at the percent biofilm coverage versus time. Unfortunately, we can't add coupons to the system. We can allow you to perfuse, for example, polymers or proteins, saliva, which contains a mixture of proteins, or some sort of inhibitory agent. We had a customer recently that was adding a monoclonal antibody to see if that inhibited adhesion and then proliferation. But things like a 5.5 5 by 5 millimeter polymer square likely can't be added. I'm happy to take any other questions at this point. Otherwise, I really appreciate your attendance, and hopefully we can be in touch soon.